Okay, so in nature there are about 300 or more known amino acids. There are really about only 20 that are used in protein synthesis, in synthesizing cellular proteins. And those are going to be the amino acids we are primarily interested in. Okay? Those 20 amino acids can be classified in two ways. The first way is the way I will not talk about today for very long, except to say amino acids can be classified as essential or non-essential. Non-essential amino acids are amino acids that we are able to synthesize. There's about 10 of them, 10 amino acids that we are able to synthesize. The remaining 10 are called essential amino acids. And those are amino acids that we have to get from the diet. Okay? So that's one important classification. That's really related more to metabolism of amino acids, and we'll get into that in the last block of this course. Okay? But I just want you to be aware of that classification. The other important classification is based on the nature of the R group. If the R group is nonpolar in nature, then those amino acids become nonpolar amino acids. If they're polar, the R group is polar, they become polar, and so on. There are two types of charged amino acids. That means the R group contains a net charge in it. That net charge could be either negative, acidic, or positive, basic. So there are two subgroups of the charged amino acids, and we'll talk about all of these actually in great detail in a minute. But before I start that, I want to remind you of these two dissociable groups we talked about, the carboxylic group and the amino group present in all amino acids. The carboxylic group under physiological pH, say around pH 7, will dissociate from COOH to COO minus. So that means amino acids under physiological conditions will have the carboxylic group in the negative, carrying a negative charge. On the other hand, the amino group of all amino acids under the same conditions, physiological pH, will be fully protonated, and they will have a net positive charge. So they will be in the NH3 plus form. And because of that, so I'm going to do a quick thing here. If you take this here under physiological pH, this H will dissociate to give you a proton, and what's left there is COO minus. So that means an amino acid has a negative and positive charge in the same molecule. And that occurs at physiological pH. And that form is called Zwitter ionic form. Okay? All right. So we're going to start with the first group of amino acids that have a non-polar side chain. I'll comfort you about all of this structural stuff in a minute, but let me get started and I'll tell you what you need to focus on as we go along. So I'm going to start with the simplest and smallest amino acid, which is glycine. So if you look at glycine here, the R group is an H. So that makes it the smallest of all amino acids. Okay? Now, I know these two things there are kind of a distraction for you. So I'm just going to say, don't worry about them right now. Okay? All they're telling you is the pH at which this proton will be half dissociated. Okay? You don't need to memorize anything. Just understand what this means, that at a pH of 2.3, this proton will be half dissociated from the carboxylic group. And similarly for the other pK2. So don't memorize, but we'll explain that quite a bit later on, probably on Friday. So glycine is the simplest, has, again, amino group, carboxylic group. The R group is H and makes it the smallest amino acid. So what makes a side chain non-polar? There is really a simple rule of thumb that you can use, which is that if the side chain or the R group is rich either in hydrogen or in carbon, C or H, then it's likely to be non-polar. Okay, that's your good rule of thumb. Okay, so what do you need to know about? You need to know the name, glycine. You need to know that it's non-polar. You need to know the three-letter abbreviation for glycine, which is the first three letters. So that gives you three things. The name, obviously, of the amino acid, the abbreviation, and what group it falls in. It's a non-polar amino acid. We move to the next one, which is alanine, and look at the side chain or R group of alanine. Alanine has a methyl group, again, rich in carbon and hydrogen, non-polar. Again, alanine is non-polar. The three-letter abbreviation is ALA, first three letters of the name. Valine, much larger R group. Again, everything that's highlighted here is the R group. An isopropyl R group, 3 carbons, valine. This is sometimes referred to as a branched chain amino acid because you see the branches here. This and leucine and isoleucine. So again, this is non-polar and its valine contains 3 carbons in the side chain. You do not need to know the structural details. Okay? I'll say that probably a half a dozen times today because I always get these emails insisting that they want to learn the structure. You do not need to know the structural details. Just understand that valine is a non-polar amino acid and it has a non-polar side chain. Leucine is very similar. Look at leucine, has an extra carbon to valine. Again, it's another branched chain amino acid. By the way, these are very important for muscle proteins, as an aside. Leucine, isoleucine, a rearrangement of the R chain. Again, if you look at 1, 2, 3, all you see in the side chain is carbons and hydrogen. Okay? So that's why they're nonpolar. And let me add something else about nonpolar. Typically, nonpolar R groups are unable to interact with an aqueous environment, because an aqueous environment is polar. So the makeup of these R groups does not allow them to interact with the aqueous environment. So that's another important thing to remember about nonpolar side chains or R groups. And then we go to this bigger amino acid, larger, quite a bit larger than the rest of them. I want you to think of alanine, and we've added to it a phenyl group to the methyl group, and we form phenylalanine. Again, all the constituents of the R group are rich in carbon and hydrogen, makes it nonpolar. Seventh one here is tryptophan. Tryptophan is actually unusual in that it has a nitrogen in the side chain. However, there's so much more carbon and hydrogen that nitrogen contribution to the polarity is minimal. So this is considered nonpolar. One important feature about tryptophan is that it has 2 rings, for example. One important feature about phenylalanine, it has a phenyl ring. The 8th amino acid has 3 carbons and has a sulfur in that R group. That's methionine, very important amino acid. It's actually an essential amino acid. This is considered, although sulfur can impart polarity, it's shielded by this methyl group. So this entire R group becomes nonpolar. So methionine is nonpolar. And finally, the last nonpolar R group amino acid is proline. And proline, as you can see, the amino group is actually attached to the rest of the R group, cyclized to form the proline ring. And this amino group is actually technically not an amino group, but an imino group, starting with an I, because it has 2 bonds versus single bond to the carbon. Here it has 1 and 2. So it's an imino group, and that's really the exception that I mentioned earlier, that all amino acids will have an amino and a carboxy end, with the exception of proline, which will have an imino end and a carboxy end. So what do you need to know about these? You need to know first the meaning of nonpolar, that they all belong here. You need to know the name. You need to know the 3-letter abbreviation. And you need to pay attention to some important feature we mentioned. For example, we mentioned the phenyl here as phenylalanine. Tryptophan having 2 rings. methionine having a sulfur, and proline having an amino group instead of an amino. That's it, basically. Don't need to know the structural details. Okay, so this is just a little more detail, but in general, everything after the alpha carbon, if you had an alpha carbon, it would be right there, right there, right there. Everything following it would be beta, gamma, and so on. You can go up to epsilon at the end. That's how you sometimes, but you don't need to know that detail. Okay, so here are some important things about all. Yes. Sorry? No, I'll get to that. The symbol. No, we're not. You don't need to know that, but I'll explain that when we get to it. Okay, so the important things about these nonpolar side chains is first, because they're nonpolar, they can't give off protons and cannot accept protons. And because of that, they cannot interact with an aqueous environment, and that means they cannot form hydrogen bonds. The only thing they can form is something called hydrophobic interactions, which means they can interact with like side chains. So one, for example, an alanine can interact, the alanine side chain can interact with the valine side chain. But it cannot, neither one can interact with water because they are nonpolar. That's what's called hydrophobic interaction. A methyl group, say, interacting with an isopropyl group, or a methyl interacting with a methyl group, and so on. So you want to think of these amino acids. I'm going to say 95% of amino acids in the body are part of proteins. There's a few free amino acids in the plasma and the cytoplasm, but really the majority of amino acids are going to be part of proteins. And the nature of their R group dictates how, or dictates their disposition in specific environments. Okay? So I'm going to go here to give you two distinct examples. The first example is this one here. I want you to think of this soluble protein. This could be serum albumin, a very abundant soluble protein in the serum, in the plasma. You have two types, and here's the entire polypeptide. And think of it as, almost like a ball, like wrapped around itself in a globular form. And it's made of two types of amino acids that make this polypeptide. Polar are red squares, and nonpolar are the yellow circles. So as you can see, the soluble environment of this protein necessitates that it exposes the polar R groups. Those are the R groups that can interact with the aqueous environment to the outside, whereas the majority of the nonpolar amino acids in that polypeptide, remember, this is a polypeptide. These are not free amino acids. This is a continuous polypeptide. Okay? Make sure you understand that. These aren't free. They're all connected through peptide bonds. But in this disposition, these non-polar amino acids are shielded away from the environment because they cannot interact with water. So in a soluble protein, you're going most likely to have polar residues on the outside, non-polar residues tucked inside that protein because they aren't able to interact with the aqueous environment. Okay, you with me? Okay, now we're going to go to a different conformation. If you have a membrane, plasma membrane, I'm going to give you this here as an example of a cell surface receptor. We're going to talk about some of these extensively in this course. And this is one type of a cell surface receptor that can interact with the environment. I want you to appreciate that the cell membrane itself, the interior of the cell membrane, is made up of fatty acyl chains, which are non-polar in nature, hydrophobic. On this side, there's polar, there's aqueous environment surrounding the cell. The cytoplasm on the other side is also aqueous. But the membrane itself, the interior, is non-polar hydrophobic. So look at this protein there. Here's another protein. Let's call it a receptor protein in the membrane. All of the hydrophobic non-polar residues are embedded in the membrane. These are non-polar. The membrane itself is non-polar. They can interact. So like interacts with like. Non-polar can interact, residues can interact with the non-polar nature of the cell membrane. Okay? I want you to understand that, and that really the R group can determine the disposition of proteins and their orientation in their specific environment. Okay, you're with me? Okay, good. Okay, proline. Proline, as I mentioned, is the only amino acid that has an imino group because it's a secondary nitrogen versus primary nitrogen, and so that's an important thing to remember. That's a unique feature of proline.